And the last speaker of the session is Andrey Kruglov here from the Rheumatism Research Center, and he's going to talk about microbiotics and how they mimic SARS-CoV-2.
Yeah, so I try to provide maybe one more explanation for the phenomenon that I just asked. So maybe it could be also bacteria which influences. So basically, it's during these 5 years I selected you the story which actually taught us something, and for me personally, and I hope you as well. So basically, how the antibody response, the mucosal antibodies in particular, can interact with the microbiota, and how you can influence one way or another either antibody responses to microbiota via molecular mimicry. And just a short introduction, because nobody talked nobody talked about microbiome before. So basically, the humans, of course, live not in a sterile environment, and we all have a lot of bacteria inside and outside, and also other viruses, fungi, and so on. And basically, if you estimate the microbiome, it's around several hundred to 1,000 species, and actually the number of bacterial cells is 1:1 to the host. So it's not, some people suggest it's like, could be the distinct organ, so it's rather distinct human, which is lives in us. And not surprisingly, when we actually get sick, then also our microbiome is changing and vice versa. So the association between changes in the microbiome and diseases is important. So which actually makes research very interesting, and a lot of people do it. And basically, when the pandemic started, a lot of people started to analyze microbiome, and soon after they stopped doing it, because as always happens that they normally just sequence, and then they find certain associations and say, well, it's associated with this type of infection, with that type of infection, but they always pick up the different bugs. And I think the problem here is that if you look into the genetic information, so the humans have roughly 22,000 genes, but the microbiome is way far diverse. It basically have several millions of genes, so it has the same amount of cells and huge genetic diversity. So, and basically, what this huge genetic diversity can be transferred in the huge antigen reservoir. So basically, this bacteria can interact with our immune system and induce immune responses or modulate immune responses. And another problem here is that this genetic information, in contrast to humans, is highly diverse, so it's constantly changing. So basically, and one of the consequences of the antigen diversity is basically that in some cases, what you can get certain molecules which resemble your host proteins, and then with additional factors like genetic predisposition or diet. So what you can get your T and B cells respond to this antigen and induce cross-reactive immune response, which can induce autoimmunity. And there are multiple cases, and basically the list is expanding first to show which bacteria and which viruses can induce autoimmunity. So, and basically, of course, it could be a pathogen and commensal bacteria. So that basically our hypothesis was that maybe there is also microbes in the case of COVID, which produce certain molecules, in this case proteins, they stimulate the B cell responses, and then basically these cross-reactive antibodies, B cell responses, basically can inhibit the pathogen. So, and of course, this can happen by molecular mimicry. So what we basically wanted to find this bacteria and to understand, okay, whether indeed this is true. The problem, as I said, it's so diverse and so big, so it's basically like to look for a needle in a haystack, which is almost impossible unless you're immunologist, basically, and you can just bring a magnet and try to get your needle outside. And for immunologists, of course, the magnet is antibody. So, and we were lucky to collaborate with Harald Cruz in the lab in Charité, who actually very early in the pandemic generated antibodies from a COVID patient. So, and what is important for us, so he purified antibodies which are reactive to RBD, and they neutralize the virus. So we wanted to find bacteria which actually can have a mimic RBD and can have a neutralization capacity. So then basically what we did, so we took a microbiota from normal healthy individuals, we stained with antibodies which recognize viral RBD, and basically you can see that indeed some of the antibodies bind distinct bacterial species, and yeah, so it's quite diverse, but basically the fact that indeed these bacteria exist and these antibodies can cross-react. So the next, basically, to understand the diversity, so we did a 16S sequencing, and as I told you, actually doesn't really result so much to the something meaningful. So more or less, we also got certain diversity, which is basically we got this fraction of antibody. I don't fully understand how it may work here. No. So basically, we sorted the bacteria and then sequenced, and as you can see, actually, we got a lot of different bacterial strains, but of course, the binding may not be specific, and this led us to further generate the single cultures from the sorted bacteria and to understand basically whether indeed this is a molecular mimicry and whether that the interaction is specific. So we cloned number of bacteria, so then we focused on 3 of them for reasons that actually, so they, on the Western blot, they showed certain strength, certain bands showing specific proteins, and they were easier to handle further in vivo settings, so basically Streptococcus salivarius and 2 bifidobacterium strains. Then what we did, so whether we can actually induce cross-reactive immune response. So then basically what you take naive mice, so you feed them with bacteria, and then you analyze response against RBD of SARS-CoV-2. And even though you fed mice with bacteria, so that you are able to induce IgA, which is against RBD or spike protein of SARS-CoV-2. So indeed, in vivo, you can generate cross-reactive immune response. Then if you take these antibodies and you analyze them for the binding, for the inhibition of binding of RBD to ACE2, you see that actually some of the antibodies which were induced are able to inhibit the binding. Then to further prove that it is a molecular mimicry, so in terms of we did a Western blot, so you can see that there is a protein which is recognized in case of Streptococcus salivarius by RBD antibodies. So mass spectroscopy showed that it is basically putative dextransucrase 01730. Then we further proved it that indeed, basically, if you overexpress the protein that is still in E. coli, you still are able to detect it by antibodies. Then if you look for molecular mimicry, so you take the antibodies which you induced in mice, which you fed with bifidobacterium or streptococcus, and then you do basically epitope mapping against viral peptides, and you see that basically these bacteria, 2 of them induce the antibodies which bind to the receptor binding motif of RBD. And to further show that indeed this protein induces, so what you do then, we immunize mice with the protein, and again we got an antibody response against RBD-OS1. Then just to show that these antibodies can inhibit the viral, so we took these mice, we did a hybridomas, we isolated antibodies which are specific to bacterial protein, and then we showed that it binds also to RBD, and it also can inhibit lentivirus entry to to the cells which express ACE2. So far it works. Basically, so this bacteria can provide a certain help in terms of infection. So what we noticed before we did actually the infection experiment, that if you use one strain, it's not so, the response is heterogeneous in terms of neutralization. So sometimes mice get neutralizing antibodies, sometimes don't. But if you mix three of the bacteria, so Streptococcus and two Bifidobacterium, so we constantly get the neutralizing antibodies. So we decided to go for this bacterial mixture, and basically then we immunized mice either with the Russian vaccine. That time it was still possible, and then with the bacterial mix, and infected them with the SARS-CoV-2. And basically 7 days later, we saw that vaccination with the bacterial mix helps to clear the virus. So it suggests basically that antibodies, cross-reactive antibodies, which are induced by bacteria, also can help in vivo to clear the viral infection. What is also here interesting that so this Covivac vaccine, the Russian vaccine, was just chemically modified, chemically killed virus in a way, and it's also induced cross-reactive antibodies against Streptococcus salivarius, which then we thought, okay, that if this vaccine can induce the anti-Streptococcus salivarius, maybe also when we vaccinate humans, we also get the antibodies and somehow modulate the microbiome. So in that case, we went already to mRNA vaccine, which is, and then what we did, so we took naive people, and during first vaccination, a second vaccination, and every 7 days we analyzed blood, stool, saliva for the antibodies for microbiome, and for everything, basically, to understand whether there is a correlation in terms of development of immune responses and microbiota composition. So, and I don't show the data, but basically in the fecal samples, we haven't seen any induction of antiviral or anti-spike antibodies, and we also didn't see any changes in the microbiome. And here in the saliva, what we see basically that the antibody, IgG antibody, starts to appear 3 weeks after the first immunization. Basically, they really boosted with the second vaccination. And if you look now into oral microbiota, so the changes starts to appear at day 28. So this is basically principal component analysis. And if you look which are the changes, the biggest change here is the Streptococcus, so that the bacteria which we cloned as an interactor with the anti-RBD antibodies. And if you further look deeper, which strain is it, so you can appreciate basically that the Streptococcus salivarius starts to grow when the antibodies, anti-spike antibodies in the saliva appear. So suggesting to you that actually that there is a correlation between the appearance, at least the correlation between the appearance of anti-spike antibodies and Streptococcus salivarius. So then okay, so if we now, the final question what we had, if we now increase the Streptococcus salivarius in vaccinated people, what will happen with the salivary antibody responses? Can we actually amplify it? And for that, basically what we did, so we fed humans with one of the available strains, the K12, basically for 2 weeks, and there were the people who were not treated. So you can appreciate basically here the absolute levels, here the relative increase, so that 2 weeks of treatment with a probiotic basically increase the levels of anti-spike antibodies, IgG. So the IgA response was not significantly different. In some cases, we saw the difference, some increase and some net. So it basically doesn't really change so much. What is important is that this increase in IgG antibodies also correlates with with a detected molecular mimicry site, meaning that receptor binding motif, and also these antibodies were cross-reactive to the other variants, including Omicron and some related SARS-CoV viruses. Yes, so basically there are also other papers which suggested it, which is good because since, as I said, microbiome research is so diverse, it's actually quite good to be scooped, or at least to see the papers which show exactly the same. And here the people studied how the microbiome affects severity or correlates with the severity, and they found basically that the oral microbiome and the Streptococcus is the top candidate for to predict a mild disease. And actually what is cool that when they try to identify the genes which are responsible for this mild course of COVID, so they found actually the gene which we cloned using the antibodies, which is actually molecular mimic of the RBD. So, which brings me to my summary. So I hope what I showed and what we learned during the pandemic, the COVID-19, that actually that there is a cross-reactivity in terms of well, antibodies in the bacteria and the viruses. So in terms of vaccination, once we vaccinate, we by provision of specific antibodies, we can modulate certain bacteria. And this bacteria also by oral supplementation can increase actually the mucosal levels, the levels of antibodies which are needed to protect from infection. So, and yeah, so of course we did a lot of studies, so that most of the studies were done before the war started, so that basically I did work with Russians and also with a lot of Charité. I'm also Russian, by the way, right? So, so the Charité and DRZ, the main basically driving force was Marina and Pavel in terms of bioinformatics. And yeah, I would be happy to discuss what we learned, and thank you.
Thank you, Andrey. Questions? So does that only work with antigens and epitopes that are on the surface of the microbiota, or does it also can be antigens that are inside? I mean, then you don't have this antibody binding, right?
Yeah. So that of course this is the way how you get the bacteria. So I mean, and of course that then I mean, it may work also with intracellular antigens, but
then it's hard to show.
We didn't do it. Yeah. So we didn't do it because, of course, you need a certain screening platform, and we use it. And for us, it's easier to do it on the cell surface.
And is there so by mechanism, how do you get immunized by your gut microbiota? Is it an accident? Do you have to have a certain disease that you get exposed to a larger amount of the bacteria, or is it just some constant exposure that we have, and it's all bacteria, or just a fraction of them?
Yeah, so in a way, so you get very polyclonal response because you constantly sample bacteria, and then basically the sampling then induces certain immune response. So then the dendritic cells present and so on. So that's kind of the mechanism, of course, and that's why it's so diverse and that's why it's stable. But once you start shifting the microbiome towards one direction, so you start to shift also immune response, and this is where the problem can start also in terms of the how you can get the other bad part of molecular mimicry in terms of autoimmunity.
Winfried.
So it's a very interesting story, a very interesting talk. So it is actually a way, there's another type of antibodies which is actually built up and is supposed to be directed against the microbiome. These are the isohemagglutinin. So which when you are born, you do not have isohemagglutinin in your blood, but then it takes years, actually, several years, to build up appropriate titers of anti-IgA, anti-A and anti-B isohemagglutinins, depending on the blood group. So I was wondering, how is the situation in children, in 1-year-old, 2-year-old children? Do they have this kind of antibodies? Did you have a chance to look whether they have such antibodies already, or is that something which is only built up later on during development?
We started actually initial project when we started, so there was no vaccine. So, and we analyzed in a population which was not vaccinated, and then we saw that basically the IgA antibodies which recognize RBD, they are present already. So, and we normally kind of annotated them to the microbiome. Actually, of course, it's pretty difficult, but we sometimes even did kind of a blind experiment so that we can just take a samples and stain them with the Streptococcus salivarius. That was the strongest predictor. And basically, if they bind, then they also have reactivity to RBD. So this is, but in children we didn't do it, I must say.
Okay, Lai? Sorry.
Do you see IgA responses to these bacterial antigens, which could be protective against COVID?
Yeah, so as I said, the response was heterogeneous in the clinical study we did. So it's not so much. The problem is that also the vaccines here in Europe, I mean, you know that it's mostly mRNA, right? And the mRNA vaccine induces very strong IgG response, like really sky high, and very little IgA. So, I mean, it would be good also to see if you, I think it's actually, just to come up, it is an amplification of already pre-existing immune response, right? So it's in a way, it's not like you induce a novel immune response. And that's why I think you need certain IgA memory B cells, or, you know, so that you can amplify them, but there was nothing really. And as I showed you also, in saliva, we don't see any differences in IgA once we start to vaccinate with the BioNTech vaccine.
Okay.
They're just not there.
Life, final question?
Yeah, interesting results. And so one thing that is notable is that after the pandemic, what has really gone up globally is invasive streptococcal infections. So, and this is for different types of streptococci, right? So group B, group A, group B, pneumococcal. And this is way more than one would expect from waning immunity due to a few years of social distancing and masks. There are some new strains, but this is really something that's striking, and you can look it up. And right now, again, we have many young people with invasive streptococcal diseases like we haven't seen before. I'm not saying this is related, but you know, somehow, since you know one of the oral streptococcal strains came up, if in some way or shape cross-reactivity or whatever, you know, can change the way we're colonized or something. I don't know.
Well, I mean, I don't know. I don't think I will comment. No, that's fine. I will tell you privately.